Okay. Hello, everyone. It's 1 p.m. and I know the schedule is kind of busy, so let's not let's not lose the time. So Aneequa, actually today Aneequa will show the phenotype. We'll show the phenotype section, and there will be actually 4 phenotypic part that she will cover, and she will use the NanoHub apps, and today I believe she will talk about the motility, mechanics, volume, and microenvironment. Yeah, thank you.
Hello everyone, I'm Aneequa, and now we would go deep into the code of PhysiCell, and we will look into how diffusion works for PhysiCell and how when we talk about cells, so what the phenotypes have, what phenotype modules do we have, and how they work together. So that's the starting point. I will go and so. First of all, I'll introduce what it is, and then first we will cover the diffusion in PhysiCell, which is achieved through microenvironment, and then I will go into the cell phenotype, and I'll cover cell motility, cell mechanics, and cell volume. And in the next session after the break, Furkan will cover cell cycle, cell death, and cell secretion. Okay. So as we mentioned in the session 0 and session 1, Paul introduced these things. So how PhysiCell works is microenvironment is the stage where all the substrates are diffusing. So diffusion solver is actually the microenvironment. Important things for microenvironment are which substrates are there in that particular simulation, what are their diffusion coefficients, what are their decay rates, what are the boundary conditions that we want to achieve for those substrates. And all these are defined in XML configuration file, which we achieve after we go through our first module, which is in the GUI. You mentioned all the substrates, and that substrates are loaded into the XML configuration file. Then the second thing is cell definition and cells themselves. So each cell has a name and cell default phenotype. So main objective of this session is to tell you what phenotype modules we have for cells and how they work. So important thing to remember that is already covered before, but I will just repeat it again. Important thing to remember, there are 3 time scales at which things work. In PhysiCell, the diffusion, secretion, and uptake, they work at 0.01 minute. So it is updated every 0.01 minute. That is the diffusion time scale for us. Cell movement, which is basically cell mechanics and cell motility, that goes at 0.1 minute. And rest of the phenotype changes, for example, cell cycle and cell volume, those go on 6 minutes. 6 minutes, every 6 minutes they are updated. So these are important things to remember. If you want something which is more fine-grained, you would have to remember where to add that into your simulation so that it goes according to this. The main purpose of these stages are just to make sure that our simulations are quick. This is to avoid a computational cost. That's the only main reason for this. Okay, so first of all, we have microenvironment. Important thing for microenvironments are only 2. One is what is the boundary conditions, what are the substrates, and what is their diffusion length. So for boundary conditions, by default, if you start, you have Born-Newman boundary conditions, but you can also have Dirichlet boundary conditions achieved. And for Dirichlet boundary conditions, we also have a fine-tuning available in PhysiCell where you can say that on one side, there's one Dirichlet condition, and the other side of the domain, there's another Dirichlet condition, different Dirichlet condition for different substrates, or Dirichlet condition for one substrate and one Newman condition for rest of the substrates. So you can achieve those kind of different combinations of boundary conditions if you want, but these are the 2 main methods through which we achieve it. Then there is also important thing, which is Dirichlet nodes. Dirichlet node is that you can go into at a certain point in a voxel, and you can make that node, that voxel a Dirichlet node, which would mean that on that point, the composition of that substrate will remain constant. Diffusion will go through it, but that will remain constant on that one. So it can act as a source or a sink if you want to work that into your simulation. Then for substrates, we have to have some diffusion rate constant for each substrate and decay rate that will define the diffusion length scale for that substrate. This is an example how it shows in the XML file. You would not directly change it. You will get this XML file once you make the GUI quick builder, and it will create that for you. But if you want to change it later, you should know how it look like. For example, in this particular example, there is oxygen and there are units. ID is 0. ID is what is the substrate ID in the system, and the system access each substrate based on its ID. And then we have diffusion coefficients, decay rates, again per minute, and micron scale per minute. Initial condition is what will be the initial condition in the whole of the domain. And then there are Dirichlet boundary conditions. So by default, Dirichlet boundary conditions are false. It means that it will be one Newman boundary conditions. But if you turn them on, then you have an option to turn them on for some of your boundaries or all of them. And you can have different values for different boundaries if you want them to do it like this. Okay. So next, I would share my app with you, which is, and we have made some apps for all of the modules that we are discussing here on this session and the section after this. And these apps are available on NanoHub. You can go to NanoHub and explore these apps. The main purpose of these apps is that these will help you explore different parameters of each of this section, and you can see that how these different parameters affect the performance of the performance, or how they change the behavior of the system. So if I go to the microenvironment, we have made this a small app. You will click on the launch tool. You will go on this page, and here it is about the introduction, what is there in the app. Then the configuration basics, we have the domain size, and right now we can only have 2D simulations in this, so it is Xmin, Xmax. Then microenvironment, since this was a microenvironment app, so we had two chemicals. One is oxygen and the other is chemical A. So suppose I turn on the Dirichlet boundary conditions for it, and at Xmin it is 10 and Xmax it is 100. Initially it is 38. So we'll see how it works. Then for user parameters, we can make a Dirichlet node. We would have to give the XY positions, which is 100, 100 at this. I will just select this, and we will make it the Dirichlet boundary condition for oxygen. And at that point, I want the concentration of oxygen to be 100. And then I run this simulation. Once you run it, the output will come here, and the results would be there in the output. And if I run it, you can see. So now this is the output for you. You can see, I do it. You can see that on the one side it is 100 and the other side it is 10. X max and X min, I put the Dirichlet conditions on that. And then at 100 point, there is a source which is 100 at this point. Oxygen concentration is 100, and these are how the bands are formed. And it will remain like this because there is no cell in the simulation and nothing is being absorbed or nothing more is being generated. So once it goes to this equilibrium, it will remain at that equilibrium stage. But in real simulations, you will have more things which would be consuming the oxygen, or there will be some other cell which would be creating something. And you can have some complex situations here. You can also see the chemical A, which has something like this. And I have to check what were the conditions for that. It has a decay rate, and its initial condition was 1. So it is very minute amount. You can see this very small amount, and that's being displayed here. And we can change it and we can look at the other stuff. So that's for the microenvironment. If you have any other questions for microenvironment, you can ask them now, or we can go to Phenotype after this. Any questions? Okay. Now we come to the cell phenotype. So cell phenotype is how we define our cells, which are the actors, which are performing all the things in our simulations. So phenotype, the modules we provide for the phenotype are cell cycle, cell death, cell volume, cell geometry, mechanics, motility, secretion, intracellular, and molecular. You can check all those in the user guide. Cycle, depth, these will be covered in the next part of the session. I will cover volume, mechanics, and motility. You will cover intracellular in later part of the week where it has some more details introduced into it. So we will not cover it in this phenotype introduction. So right now I will go into the motility, mechanics, and volume and how they work in PhysiCell and what they mean when we set them different parameters for them. So first of all is motility. We use motility to actually show the cell movement, and cell movement in a biological simulation can mean many things. It can mean a chemotaxis towards a thing. It can be chemotaxis away from another chemical. It can also mean just random movement into the cell is just randomly moving into the environment. Maybe it's a you want to perform a bacteria which is randomly moving into the environment, or you want to simulate a immune cell which is randomly moving in the blood, and then it finds something and then it starts moving that towards that. So all these concepts can be modeled using motility part of the phenotype. So important parameters for the motility are: first is migration speed, how quick or how slow you want your cell to move. Then this is bias direction. Bias direction is basically your chemotaxis direction, which chemical you want to move towards, and that will give us the bias direction. And then we have migration bias value. This migration bias value is that how much importance is given to that chemical. So if it is 1, it would mean that whatever the gradient of that chemical is, it will just go into the straight line towards that gradient, in that gradient. And if it is 0, it will just ignore the gradient and it will keep moving the Brownian motion, random motion completely. For reality, we want something between around 0.5, where there is some stochasticity into it, but still overall it's going into one direction. So that would be a more realistic value for migration bias. And then there is persistent time. Persistent time is how often cell goes into the one direction before it checks again for the gradient. So again, persistent time is for computational complexity. If you have a lot of persistent time, it will go into one direction, keep going into the one direction before checking again, and then changing its direction, then changing its direction. And if the persistent time is really small, it will take small steps. So it will check again. So another important thing is there is another parameter which is motile, which is for every cell. So each cell phenotype has a parameter is motile. It has to be true for motility to work. If it would be false, no matter what the speed is, cell will not move. So you have to have cell is motile true first, and then we use 2D to true if it is a 2D simulation, because we want it to be in 2D. And then we use the substrate for the chemical, whichever chemical we want chemotaxis towards or against, we use that. And direction 1 is to go up the gradient, direction -1 is to go against the gradient. And again, this is the important thing: if substrate doesn't match something defined in the microenvironment, the initialization will fail, because the substrate should be defined in the microenvironment which substrate we are using for the motility. Again, we have an app for this. We can go to app and you can see that. All these apps are available. You can just go and explore them on your own if you want to explore them. So if I run this by default, which is user parameters are, there are 3 number of cells which are motile and which have a persistence type of 30, migration speed of 2, and migration bias of 0.8. So 0.8 is a big migration bias. So you would see they are moving fairly in a straight line. It's not exactly the straight line, but migration bias was strong, 0.8. So they are almost going into the straight line. A little bit of changes in that, and you can see if I make my migration bias to 0, it would mean that they will just be randomly moving. They would not move into the direction of the bias. So if I run it and we put again a new outputs are being generated, you can see here. See, this one is going down, this one is going up. So they are not going into the direction where we want them today because we set the migration bias to a 0. So they are basically they are Brownian motion. They are randomly moving into the domain. So depending on your simulation, you would decide what migration bias, what migration speed works for your application. Is there any question here? I can't see the chat right now. Okay. Can't do I need to answer any question?
So right now, actually, no, actually, like I'm just answering questions through the chat.
Okay.
So one thing is maybe to mention is actually we can set the bias direction based on the substrate gradient, and which means the chemotaxis, and you can do both towards or from, and so yeah, you can use the direction, yeah, chemical direction.
But that's for the motility. That's the first part of the phenotype, cell phenotype, which is implemented by the PhysiCell, that is available for you. So it is just like out of the box chemotaxis for you. If you want to implement chemotaxis for your cells or the random movement, that's where you will go and implement that. Okay. The next thing is mechanics. Again, mechanics is something which is basically the cell to cell. So it is PhysiCell is a physics-based model. So it is like how much adhesion is between the cells and how much repulsion strength is there between the cells. The default model uses the partial potential functions. You can have your own potential functions if you want to implement, but right now we have potential functions which based on adhesion and repulsion strengths, they get equilibrium distance. So when there are a lot of cells jumbled up, so if there's like one point, you make a lot of cells at the one point, they will repel each other and they will arrange themselves, because they would not be two cells at one point. They will be repulsion, and there's some, you can control the adhesion, how close you want the cells, how distance you want the cells. So this is the basic purpose of the mechanics. It is not, mechanic is not for motility, it is for just a stable environment where cells would be. And again, mechanics also tells us that even if the motility is off, if you initialize the cells very packed together, they will get a repulsion in the beginning and they will arrange themselves. Okay. So important parameters for mechanics. Mechanics for important parameters are adhesion strength. Adhesion strength is how it is between 0 and 1. So 0.4, right now it is 0.4 here. Then there is repulsion strength, how much micron per minute, how much repulsion is there. And then maximum adhesion distance is that how far beyond the cell nucleus the strength effect of the adhesion or repulsion will go. So if two cells are very far apart, they would have no effect on each other. And if they are very close together, there would be some effect there. Then there are some functions that are provided by the system. For example, set relative equilibrium distance and set absolute equilibrium distance. So self-relative equilibrium distance is relative to the radius of the cell. So it will set the distance according to those distances. And for cell absolute equilibrium distance, how much distance we want between the cells. And normally we kept it between double the cell radius. So it is like cells are just next to each other. The thing you need to make sure here is that we change cell adhesion, we need to change cell repulsion, but that's not what we recommend you to do. We recommend you to use these functions for changing that. And that we will recommend you to use for if you want to change the distance between the cells. but still you have the available for change adhesion strength, repulsion strength, and maximum adhesion distance. Okay. I will go to the app for that. We have an app for that also. Sorry, I think there are some questions in the chat.
Yeah, so actually Zoe's question is related with the microenvironment, and she is asking about can we change the diffusion coefficient in the middle of the simulation, or is it the same through the all simulation? So actually you can change it, but
why would you change that?
Yeah, so you can change it, but the thing is, in BioFM you cannot define the diffusion coefficient specific area. So if you change the diffusion coefficient, it will just affect all domains. So so right now it's not implemented yet. And Daniel has a question actually about this mechanic. Cell adhesion is between 0 and 1. Is it the 0 and 1? And also has units micron per minute. Is that correct?
I would have to check again. I think this is correct. Adhesion strength is between 0 and 1, and repulsion strength is 10. I'm not sure about it. Furkan, can you?
If I may?
Yeah.
I'm sorry, Furkan, if you would like to address this, I'm sorry. The adhesion strength can be any number. It doesn't have to be between 0 and 1. And what ends up mattering a lot more is the ratio between the adhesion strength and the repulsion strength. If you look into the mechanics, the detailed mechanics model in the PhysiCell paper from 2019 in computational plus computational biology, it gets into the details of, it's kind of a Lennard-Jones-like potential, if anyone's familiar with that, and it's similar to because I'm on the spot, I'm completely blanking on the name, like, I'll think of the name as soon as I'm done. But it's similar to other physics models where you kind of have squishy balls running into one another. And then as far as the units, I had thought that it was unitless, but I can't speak to that definitively. I'm sorry.
Okay. Yeah, thank you, John. Thank you so much. Yeah, I would recheck that. Yeah, maybe I made the mistake. It is not between 0 and 1. It is just 0.4 by default, but maybe I got this impression from 0.4. Okay, so this is the cell adhesion strength, cell repulsion strength, maximum adhesion distance 1.25. Right now I'm not using any of these functions, and I will just go with the default, and I will run this simulation here. And once they start generating, you can see, yeah. So if you see at 0.0, we did it on purpose. We initialized the cells in this way that right now and at this point there are two cells. They are just overlapping each other. They are a little bit at distance from each other, but they are still overlapping. These cells are just at equilibrium, like almost at equilibrium, and these cells are so far apart that they will not interact with each other. So when we run the simulation, you would see that these cells are repelling each other because they were at the same point. So they started repelling each other, and nothing happened to these ones because they were already in a good distance. These two, they are not basically interacting with each other, and these are already at the equilibrium distance, so they are there. And we use some extra interface to actually represent these light blue circles. These light blue circles represent the adhesion distance. And normally when you will use a out-of-box PhysiCell simulation, you would not get these extra circles there. that we use just for the purpose of the simulation to actually demonstrate what the adhesion strength is. And here we can show you, for example, if I change the adhesion distance and make it really big, like 4.5. So now its adhesion distance is really big. And then we run this simulation, and I'll go back and see now the adhesion. Now you can see the interaction distance is a lot more because they can, this is 4.5 times bigger than their nuclei. And when I will run this simulation, you would see that all of them are interacting with each other and they are coming closer because they are at distance, and then they will come even closer. Okay, they have found two different groups, and they have arranged themselves in this way. So it is important to note that these things can get real complex real quickly if you change, for example, adhesion distance here, that which cells which were like two cells neatly arranged, they are just interacting with each other. And maybe if I run it longer, it can even interacting between these two cells, these two clusters also. So this is important to remember. So I will change it back to 1.25. And then I want to use some relative equilibrium distance. You can use idea of this function. For example, relative equilibrium is 2 times the cell nuclei, and we'll run it. And you can see the output. Again, it started from here, and these ones repelled each other. And this would like the first one because 2 times, that was the equilibrium distance that we were hoping for in the first set of the parameters also. So that's there. If you want to make it even bigger. Okay, now they are moving, but they are moving really slow. And that's why you cannot see it. If I run it longer, you might see it. Okay. So any questions here so far?
Yes, Robert is asking, can you add multiple cell phenotypes with different adhesion distances?
Yes. Each cell phenotype, when each cell definition has different, like all the parameters I'm discussing in this slide, other than microenvironment, each cell has its own mechanics. Each cell definition has its own mechanics values, its own motility values, its own volume values. Everything is particular to that one cell. So if you want 2 cell types, one cell type with a lot of adhesion and the other cell type with a lot of repulsion, yes, you can do that. And you can have like 10 cell types with different adhesion and repulsion strengths. You can have that. Any other question?
So actually, I kind of like skipped the Connor question. Actually, he was asking about the movement and also the position of the cells. And he was asking, is actually like, sorry, I just want to look again. If the cells are modeled as off-lattice center base, are they modeled as points on mutable mesh to define cell-cell interaction like other softwares? How is velocity implemented? So actually I skipped this one because it is kind of like coupled with the mechanics. So the motility and those kind of like forces and adhesion and repulsion is kind of is calculated in mechanics time step. They are kind of mutual actually. So therefore they are at a kind of like a repulsing and making adhesive forces, and also the velocity is adding a kind of like force to the cells. Therefore they can just move off lattice.
Yeah, and the thing is, this is our lattice model. So your diffusion is at a different. It's like in the microenvironment, and cells are on the phenotype, and they are with respect to microenvironment. They defined their nearest voxel in the mesh, which voxel they are in. So this is how we define them. Okay, that's for mechanics. I will move to my slides
And
again.
also, actually, Robert has a question. How are these parameters are identified? Are they retrieved from literature or fitted?
It depends on your problem and it depends on your simulation. So if you have these, like if you have data for these kind of parameters, you can use them. Or if you have, it depends on the problem, how, or sometimes it is just a parameter fitting where you want to achieve how close or far apart your cells are in the natural environment, and you want to replicate that in your simulation, so you can fit different parameters. It depends on your problem. We don't have any recommendation for anything. It's just depend on the problem. Next, the last part in my in this session, that is the volume. And volume is, I hope you can see all of it. You see? Okay. So volume records in PhysiCell, volume and geometry are linked with each other. So it is important that you keep those in things in mind. I am not covering geometry here because geometry is not actively actively updated using volume. It is not updated directly. So you don't, if you want a bigger cell, you will not change the geometry or the radius of the cell. You will change the cell volume. That's how we implement that in PhysiCell. So although there's a geometry class inside the phenotype class, you would not use that. We will recommend always using cell type phenotype volume. Now, volume is divided into two volumes: the cytoplasm volume and nucleus volume. So we have nuclear volume, cytoplasmic volume. Then we have solid and versus fluid. So we have how much solid friction we want in the nucleus, and also how much solid we want in the cytoplasm. Similarly, how much fluid you want in the cytoplasm and how much fluid you want in the nucleus. Gasification friction, and this is how this is regulated. This is fluid change rate, nucleus biomass change rate, cytoplasm biomass change rate, and dt for volume is our third dt, which is the dt which is 6 minutes. So it is updated every 6 minutes, and we change the volume and we try to achieve the target volume for each of them. Now, important thing here is that when you go into this class, you would see a lot of parameters, and you might want to change all of them, but changing most of them will not make any difference, because the important things here are to get to that target volume which we want. So target volume is fluid friction change rate, cytoplasmic change rate, nuclear biomass change rate, and what we want the final volume to be. If you will change anything else, it will just go to that target volume. So that's we don't have a specific target volume for it. We have different parts of it, and you would have to change that. So if you change the rates, it will show how quickly cell moves to the target volume or how slowly it goes to the target volume. Relative rupture volume, it is useful in death models. So I will not discuss this here, although it is part of the volume, but rupture volume is used in apoptosis and necrosis, and Furkan will cover it there. And then we have state variables and target parameters. These are important things to remember, because some of the parameters are only for the state. If you will change them, it will not change the target one. And if you change the target parameters, they will actually change. It will make the difference. I'll go to the app and it will be easier to explain using that. So this is the basic introduction. We have config basis, microenvironment. We are not using microenvironment for any of these apps which I discussed right now, like motility, mechanics, and volume. It is just there. Now these are the parameters. And if you see, there are a lot of parameters and it can be overwhelming to look at them. Important thing to remember is we have made the dividers. So parameters are the one that will take your volume to your target volume. So you want a target solid cytoplasm in these cubic microns. This is what you want. Solid nuclear, 75% fluid fraction. This is which will give you that solid thing. If you will change any of this, these are the state variables. They represent the state of that cell at a certain point. And if you will change it, it will change it momentarily, and then cell will go back to that target volume, whatever that volume is. Sorry. So these are the things there. And here, the important thing to remember is if you want a bigger cell and you want a bigger cell, just use this function, use everything multiply by ratio, because we have set the defaults to like 75% fluid fraction, which works most for most of the applications. If you want a bigger cell, multiply it with 2, 3, 4 bigger cell. If you want a smaller cell, multiply it with 0.5. This function will take care of everything. It will change every parameter according to that ratio. So it is the easiest part to make your cells bigger or smaller. First, I will run it, and you will see nothing will happen. So there is a cell, its cytoplasm, its nucleus, and it is there. Nothing is happening. It is the default parameters which were there. State variables were with relation to that, so this will remain there. But then if I change some of the state variables, for example, this volume total, I change it and I add I have added too much. So I add some zeros to it. Now my total volume is increased, but like I said, state variables will change it momentarily and cell will go back to its target volume. So if I run it, in my output plots, you see, at 0, it was, I have to do it again. So at 0, this is the initialization. It got bigger and then it's getting smaller. The state variable changed. The state changed its state at that moment, but it is going back to your target volume and it will go to that target volume which I initialized with. So if I will change these parameters, they will actually change the cell. And if I want to, I'll go back to this. 494. And if I want to double the size of my cell, I can just use this function and I will run it. And see, it's getting bigger and it's going to the double the size. Double the size of what was present in the beginning. So this is the easiest way to change your size. And if you want more fluid fraction, more solid biomass, then you can change these parameters. So please remember these parameters. Don't try to change these one, and then you will see that nothing changed and you will not get the results there. So any questions here?
So right now, no, James actually asked for the collection of the all apps, and I put the link for.
Yeah, we have collection of all these apps, and yeah, Furkan has shared the link with you, and you would be able to connect it there.
Robert actually asked a question. Are all cells circular or spherical, or can more complex shapes be created?
So right now, all cells are circular. This is by default how we define them. We define the volume, and then we define based on that volume, we consider them as sphere, and based on that volume, we calculate their radius. And they are spherical in shape, but you can, Paul will discuss some of the things where you can combine 2 or 3 cells and you can make a worm or something like this. But right now, all the cells are spherical. That's the short answer.
And Divyo actually has a question. What decides the time point at which changes made in state variable are seen?
That depends on the volume dt. So volume dt is 6 minutes. So 6 minutes of simulated time, it will recheck again and update the volume. So every 6 minutes. Every 6 minutes of simulated time, it will be checked. In my simulation, you would be saying it instantly because I was storing the information, I guess, every 30 minutes. So by the 30 minutes for pause of simulated time, it has already changed. But it will, in reality, it will change every 6 minutes. Any other question?
Those were all actually.
Okay, we are before time actually. We
Yeah.
have 5 minutes. If you have any other questions, please, you can ask them, and you can set these cell phenotype options in your GUI where you will set all of them, and then you can later change them in the C++ also. You can change them in XML also. There are different ways you can modify them, and these phenotypes models, these are different for each of the I'm sorry, I skipped something. These are the same for each one of them, and you would be able to see that each cell can have its own phenotype, and its phenotype have its own motility rate, its own mechanics, its own cell cycle, its own death rate. Everything would be there. And in the next session, after we will have a 15 minutes break. After that, Furkan will go through cell cycle, cell death, and cell secretion. Thank you.
Yeah, we will start into 2 p.m., I believe. Yes. Actually, Farnush has a last question.
Okay, I can answer it.
Does PhysiCell include default values for cell parameters, or one should set all the parameters?
Yes, PhysiCell includes all the default parameters, and you can see that you can go into the PhysiCell, you can see that in the user guide, some of them are mentioned. By default, we have used the default parameters in our apps also for easier understanding. So these are also the default values. And yes, if you will not set anything, PhysiCell will use the defaults for that.
Thank you very much all. And let's meet on 2 p.m. Eastern time.